This will be our mind map focused review session for the functions and physiology of the respiratory system, beginning with the overall functions of the respiratory structures. Keep in mind that the respiratory system's primary responsibility is the exchange of two respiratory gases, oxygen and carbon dioxide. Oxygen will be exchanged between the air in the alveoli and the bloodstream by diffusing across the respiratory membrane into the bloodstream. It's very quickly loaded onto the protein called hemoglobin that's found within the erythrocytes, and those erythrocytes will then be carried to the tissues, where the tissues will use that oxygen to make ATP. On the other hand, we have carbon dioxide, which is picked up at the tissues, converted into bicarbonate, where it is carried in the plasma to the lungs, and at the lungs it is converted by an enzyme back into carbon dioxide, where it is exhaled. And keep in mind that carbon dioxide is created from glucose. So glucose is a carbon sugar, so the carbon in carbon dioxide comes from the glucose that is metabolized in order to produce ATP energy. Looking more broadly at the respiratory system, we can classify the respiratory system structures and organs into one of two categories. Those organs and structures that belong to the conducting zone, and those organs and structures that belong to the respiratory zone. So the conducting zone structures are all of the organs and the structures that are not directly involved with gas exchange. So structures like the nose, the pharynx, the larynx, the trachea, and some of the large diameter bronchi. Because these structures, at their most basic, function as passageways in order to move volumes of air to and from the alveoli of the lungs. But they also have secondary functions, or some of them have secondary functions where they will warm and humidify incoming air, as well as cleansing the air from dust and debris. On the other hand, the respiratory zone includes the structures that are directly involved in gas exchange, which are primarily the alveoli and the associated capillaries, because those are microscopic structures that are very thin, and there's millions of them inside of the lungs. And so those alveoli represent the kind of basic structure across which the gas exchange is going to occur. Looking more specifically at the nose and the nasal cavity, we see that each nostril, both the right and the left, open into a small chamber called the nasal vestibule. The nasal vestibule typically houses hairs that function to filter and cleanse incoming air from any kinds of dust that are in them. That nasal vestibule will expand to form the rest of the nasal cavity, and that nasal cavity is lined with an epithelial tissue that's called the olfactory epithelium that functions in the sense of smell. As a whole, the nasal cavity warms and humidifies incoming air. We also see these little structures that hang from the walls of the nasal cavity that are referred to as the nasal cone key. And the nasal cone key help to mix incoming air and create turbulence in that incoming air, so that way that air spends a little bit of time in the nasal cavity to become warmed and humidified. We also have some sinuses, namely the frontal and sphenoidal sinuses, which are found in the frontal and sphenoid bones of the skull. And those not only function in warming and humidifying the incoming air, but they also act as resonance chambers for speech. We see that the floor of the nasal cavity is made of the hard and soft palates, which function to separate the nasal cavity from the oral cavity. The hard palate is a structure that's made out of bone, and the soft palate is made of skeletal muscle that is continuous with a structure called the uvula, and the uvula and the soft palate function to close the nasal cavity when swallowing. So that way food does not make its way backwards up into the nasal cavity. Next, we'll examine the pharynx. The pharynx is a passageway that's primarily composed of skeletal muscle as well as mucous membranes, and it has three general regions. We see that the superiormost region is called the nasopharynx, which is the passageway from the nasal cavity into the pharynx, and it functions as a passageway for only air, so it is strictly a respiratory system structure. Meanwhile, the oropharynx, which lies at the posterior part of the mouth, functions as both a passageway for air and food, so it shares functions with both the respiratory and the digestive system. The same goes for the laryngopharynx, which is also a shared passageway for both air and food. The laryngopharynx branches anteriorly, which is the passageway that will enter the larynx, and that will continue on with the respiratory system. And the posterior branch of the laryngopharynx is the entrance into the esophagus, which will strictly be a digestive system structure. Next, we will examine the larynx, which is a passageway that is formed primarily from hyaline cartilage rings. And the function of the hyaline cartilage here is that the hyaline cartilage resists the pressure changes that occur during the normal breathing rhythms of inhalation and exhalation. And hyaline cartilage does not collapse under those pressure changes, so it's an ideal material and ideal tissue for creating that passageway. The larynx functions as a conduction zone structure that helps to move air between the pharynx and the trachea. It's also referred to as the voice box because it contains the vocal folds, which we'll see in a minute. The epiglottis is an elastic cartilage structure that closes the superior entrance to the larynx when swallowing, so that food or beverages that we're consuming do not enter the larynx, but instead are directed posteriorly into the esophagus. The thyroid cartilage is the cartilage that composes the bulk of the larynx, with the cricoid cartilage being located on the inferior side of the larynx, and it's the structure of the larynx that connects the larynx to the trachea. We see interiorly that the corniculate and the arytenoid cartilages on the posterior interior side of the larynx help to support the vocal folds, as well as the associated muscles that move the larynx and move the vocal folds during speech production. The vestibular folds are located in very close proximity to the vocal folds, but instead function to close the space between the vestibular and the vocal folds. That is, they close the passageway in the middle of the larynx, which is referred to as the glottis when we swallow. So the vestibular folds, even though they're in very close proximity to the vocal folds, they don't function in producing speech as air passes over them. The vocal folds are located just inferiorly to the vestibular folds, and these are the folds that actually vibrate as air moves over them, and those are what produce the speech sounds. Moving inferiorly, we'll now examine the trachea, which acts as the conducting zone structure that extends from the larynx to the bronchi of the lungs. It's made of approximately 16 to 20 C-shaped rings of hyaline cartilage, and just like the hyaline cartilage in the larynx, the hyaline cartilage in the trachea helps to prevent the trachea from collapsing during the normal pressure changes that we see during inhalation and exhalation. Its innermost lining is covered with a pseudostratified columnar epithelium, which is an epithelial tissue that produces mucus in order to trap dust and debris that enters the trachea. At the base of the trachea, near a piece of cartilage called the carina, the trachea branches into the primary or the main bronchi that enter each of the lungs. And the carina, as part of the trachea, not only does it consist of hyaline cartilage pieces, but the carina also has sensory receptors located on its inner walls. And those sensory receptors are very sensitive to touch. So anything that lands on the inner part of the carina, like dust or debris or even liquids, will trigger coughing to try to expel anything from the trachea before it enters the bronchi and ultimately the lungs. Looking microscopically now, we'll examine some of the structures of the respiratory zone, specifically the alveolar sac as well as the alveoli. The alveolar sac is a structure that is really just a cluster of alveoli. And there are small passageways, microscopically small air passageways called the alveolar ducts that are composed mostly of smooth muscle that help to connect a structure called the respiratory bronchiole to an alveolar sac. Now, the alveolar duct and the respiratory bronchioles are some of the smallest diameter airways, and in particular, the alveolar ducts can change radius or diameter in order to control airflow into and out of that particular alveolar sac. The alveoli themselves are very thin microscopic air sacs that contain capillaries on their surface for gas exchange. Typical numbers of alveoli for the entire respiratory system, that is both the right and the left lungs, range from anywhere from about 300 to 500 million. Our lecture textbook, being the OpenStax textbook, has the number from anywhere from 480 to 500 million alveoli in both of the lungs, which helps to create an enormous surface area for gas exchange, on the order of about 800 square feet or so of gas exchange surface in both of the lungs because of the microscopic size and thinness of the alveoli. Looking more specifically at the alveolar wall and the respiratory membrane, we see that the alveolar wall is composed primarily of two cell types: the type 1 alveolar cell, which is a simple squamous epithelial cell that forms really kind of the bulk of the alveoli wall. It does contain a little bit of elastic connective tissue in it that allow the alveoli to stretch during inhalation and recoil during exhalation, which helps to push or squeeze some of the air out of the alveoli during the exhalation cycle of respiration. The less common type 2 alveolar cell functions to produce a detergent-like substance that's called surfactant. The surfactant is primarily composed of lipids and proteins, and what it helps to do is helps to break up the surface tension of the fluid that's coating the type 1 cells. What we see is that the walls of the alveoli and the type 1 cells have a very thin microscopic layer of water on their inner surface. The water that's in the alveoli is necessary to help with gas exchange, because the gas exchange has to happen across a surface that is damp. And we find that the surface tension of water in those microscopic structures can actually function to collapse the alveoli. So the surfactant functions in reducing that surface tension, which allows the alveoli to inflate more easily between breaths. It prevents the alveoli from completely collapsing due to that surface tension and helps the alveoli inflate more easily. We also see alveolar macrophages, or a type of monocyte, that function to kill any bacteria or break down dust or debris that enter the alveoli. We also have alveolar pores, which connect adjacent alveoli in order to allow air to distribute evenly through the lungs. And then finally, looking at the wall of the alveoli, we see that the respiratory membrane is the membrane that gas exchange occurs through. As we saw in the previous slide, it has an extremely high surface area for what it is, about 800 square feet. And it's composed of three structures: the alveolar epithelium, which is primarily the type 1 alveolar cells; the capillary endothelium, which is simply the epithelium that lines the inner walls of the capillaries that coat the surface of the alveoli. And there's also a structure that exists in between them called the fused basement membranes. And this is essentially a protein or glycoprotein glue that holds the alveolar epithelium to the capillaries. Next, we'll examine the broader functions and anatomy of the lungs. The lungs, of course, are the pyramid-shaped paired organs that are located above the diaphragm muscle and sit on the right and left sides of the heart. We see that the right lung is a little bit larger and has 3 lobes in comparison to the left lung, which is slightly smaller and has 2 lobes. The reason that the left lung is slightly smaller is because the position of the left ventricle of the heart occupies some of the space that would otherwise be occupied by the left lung. Both of the lungs are enclosed by a membrane that's referred to as the pleural membrane, and it is a double-walled membrane that has a thin layer of fluid between those walls. The parietal pleura is the part of the pleural membrane that is attached to the wall of the thoracic cavity. The visceral pleura is the layer of the pleural membrane that is attached to the surface of the lung. And we see that the pleural cavity is the space that is between the parietal and visceral pleura and contains a fluid called the visceral pleura, the visceral fluid, or the pleural fluid, which helps to reduce friction when the lungs expand and compress during breathing movements. But the pleural fluid also helps the lungs as well as the visceral pleura to stick to the parietal pleura. So the water, the fluid that's in there essentially also acts like a glue so that the lungs are able to follow what the thoracic cavity is doing in terms of changing volume during the normal inhalation and exhalation cycles of respiration. Next, we'll examine some of the basic physiological principles of the respiratory system, beginning with Boyle's law. And Boyle's law is a principle from chemistry and physics that states that pressure and volume are inversely related to one another. So this is a law that helps to explain why airflow is created into and out of the lungs when the thoracic cavity expands and increases in volume during inhalation, and then compresses and decreases in volume during exhalation. So we see that air is going to flow from high to low pressure. So it's going to flow in much the same way that materials will flow during diffusion. So that is, we saw that diffusion creates movement from high to low concentration. Similarly, air will flow from high to low pressure. So anywhere that we create a low pressure environment is where air is going to flow into, which is exactly what we see when we inhale. We see that the muscles of the thoracic cavity help to create an increase in volume, which is going to subsequently, because of Boyle's law, create a decrease in pressure inside of the lungs and inside of the thoracic cavity. And that's going to help create that initial airflow into the alveoli, where gas exchange can then occur. On the exhalation cycle, we see that the muscles will start to decrease the volume of the thoracic cavity, which is going to increase the pressure inside of the alveoli and inside of the lungs. And that's going to help to create airflow out of the alveoli during that exhalation cycle. Specifically, we refer to inspiration as, or inhalation as inspiration during the respiratory cycle, and exhalation is also referred to as expiration. What are the specific muscles that are involved during inspiration and expiration? So during quiet inspiration, which is the breathing movements that are occurring during rest, quiet inspiration we see involves only the diaphragm contracting. The diaphragm is a dome-shaped muscle that when it contracts, it will flatten, and that flattening out creates an increase in volume of the thoracic cavity. And we see that the lungs and the walls of the thoracic cavity expand as a result of that. As we just saw in the previous slide, we saw that when volume increases, pressure decreases, and that creates flow into the alveoli. Now, during quiet expiration, which is the quiet exhalation movements during rest, we see that the diaphragm relaxes, which is a passive motion. So because the diaphragm relaxes to create those expiration movements of air, we see that the quiet expiration is a passive process that doesn't involve any energy contribution by the body. It simply involves relaxation of that diaphragm muscle. That diaphragm muscle will return to its dome shape, which will decrease the volume of the thoracic cavity and also decrease the lung and alveoli volume, which increases the pressure, as we just saw with Boyle's law, creating airflow out of the lungs. Now, during physical activity, exercise, or COPDs, we see that forced inspiration will recruit additional muscle groups. The diaphragm will contract, just like with quiet inspiration, but a muscle group that is referred to as the external intercostals will also contract as well. The external intercostals help to elevate the ribs and further increase thoracic volume during physical activity, exercise, or COPD. On the other hand, we see that forced expiration involves contraction of the internal intercostal muscles, the external oblique muscle, the internal oblique muscle, and the rectus abdominis muscles, because all of these muscles help to compress and lower the ribs in order to decrease the thoracic volume, which is going to increase the pressure in the alveoli, increase the pressure in the lungs, and create airflow out of the lungs. Now, in lab, we used spirometers to measure the inspiration and expiration volumes. The spirometer is a device that simply measures the volume of air in a given breathing cycle, either into or out of the respiratory system. We saw that there were 4 volumes of interest, the first being tidal volume, which is the typical volume that's moved during normal quiet inhalation, or inspiration and quiet expiration. And tidal volume averaged about 500 milliliters for a typical healthy adult male or adult female. We also saw inspiratory reserve volume, which was the volume that was forcibly inhaled beyond tidal volume, so that would be the equivalent of taking a forced inhalation. Expiratory reserve volume was the volume that was forcibly exhaled beyond tidal volume, which would be equivalent to a forced exhalation. And we also saw residual volume, and residual volume was the amount of air that remains in the lungs and in the alveoli after a forced exhalation. And this is the volume of air that remained in the alveoli to keep the alveoli inflated between breaths. Because if the lungs completely emptied themselves of air between breaths, the alveoli would actually collapse, and it would take a lot of effort and take a lot of energy to reinflate them, mostly because of that thin layer of water that lines each alveoli and the surface tension of that water. That surface tension of that water would cause the alveoli to collapse completely. But the residual volume remains in those alveoli to keep them inflated between breaths. Now, we can't measure residual volume directly, of course, because it remains in the alveoli even after the most forceful of exhalations, but we can estimate residual volume using 2 equations, and those equations take into account 3 variables. And those 3 variables are gender, so that's why we see the 2 equations. We have an equation for males and we have an equation for females. Each of those equations also takes height and age into account as well. We also see that there are 2 respiratory capacities of interest. One of those is called vital capacity, which also goes by forced vital capacity, which is defined as the amount of exchangeable air. So vital capacity is a forced inhalation followed by a forced exhalation, which takes into account tidal volume plus inspiratory reserve plus expiratory reserve. So we would simply add those 3 volumes together in order to get vital capacity. And finally, we have total lung capacity, in which we add residual volume to vital capacity, which means that total lung capacity is simply the sum of all 4 of those volumes of interest. Typically, it averages about 4,200 milliliters for a healthy typical adult female, and about 6,000 milliliters on average for a typical healthy adult male. We see that one of the variables that promotes airflow to and from the alveoli is resistance of those airways. So in much the same way that we saw that resistance to blood flow was created and was explained by Poiseuille's law. So resistance to blood flow was created because blood is viscous and sticky, and so when it flowed, it had a tendency to stick to the walls of the passageways that it passed through. Air also is viscous and sticky. It is not as viscous and sticky as blood is, but air still encounters resistance as it passes through the respiratory pathways. And so therefore we can apply Poiseuille's law to airway resistance. We see that airflow is created by a change in pressure over resistance, just like blood flow is created by a change in pressure over resistance. And we saw that airway radius or diameter, just like with blood flow, plays an important role in altering resistance, because we see that resistance is in the denominator of some of the derivatives of Poisson's law's equations. And because resistance is in the denominator, means that anytime that we have a small resistance, that is, anytime resistance goes down, airflow will increase. On the other hand, if we see that resistance becomes large, then because it's in the denominator of the airflow equation, anytime resistance is large, airflow decreases, because we're dividing that change in pressure by a larger number. So we see that an increase in the radius of an airway will decrease the resistance. It'll help make resistance smaller, and again, because resistance is in the denominator, anytime that resistance is small, airflow will be great, that is, airflow will be large. But on the other hand, if we see that airways decrease in radius or diameter, that will function to increase the resistance to that airflow, and it will inhibit or decrease airflow as a result of that. Conditions like asthma will create inflammation and spasms, particularly in the smooth muscles of some of the small bronchioles. Asthma is typically triggered by environmental factors like inhaled irritants or like dust, pet hair, smoke, and things like that. But asthma can also be triggered by exercise or stress. And we see that primarily what happens is in those spasms and inflammation of the bronchioles is that it reduces the radius of the airways. That functions rather to increase the resistance of the airflow through those airways. So by decreasing airway radius or diameter, we increase airway resistance and decrease airflow through those airways. Last but not least, we will examine a principle of physiology that's called Dalton's law of partial pressures. And Dalton's law of partial pressure states that the total pressure of a mixture of gases is equal to the sum of the partial pressures of each gas within that mixture. We see that there's two primary gases that make up Earth's atmosphere. The first is nitrogen gas, which is abbreviated N2. Nitrogen gas makes up about 78% of Earth's atmosphere. And that gas is largely kind of ignored by our physiology. The nitrogen gas that we inhale and exhale regularly on a typical basis really doesn't have a huge effect on our physiology. Simply what it helps to do is contribute to air pressure. The other gas that is of significant quantities in the Earth's atmosphere is oxygen gas. Oxygen gas makes up about 20.9% or 21% of Earth's atmosphere. And the air pressure that's specifically created just by the oxygen gas is referred to as the partial pressure, because we see that air is in the atmosphere is primarily a mixture of nitrogen gas and oxygen gas. And so the pressure that's created by each of those gases is what we call the partial pressure. And so the partial pressure of oxygen is what we're going to mostly focus on here. And partial pressure of oxygen is denoted as PO2. The diffusion or the movement of oxygen into the blood is partially dependent on the partial pressure of oxygen, with higher partial pressures of oxygen functioning to push more oxygen or help more oxygen diffuse into the bloodstream, with lower partial pressures of oxygen resulting in less diffusion of oxygen into the blood. So we see that as a whole, both nitrogen and oxygen gases are gases that exert pressure on surfaces, because nitrogen and oxygen are both molecules, and because those molecules have mass and weight and are moving, they exert pressure on surfaces. So the more molecules that we have, that is the higher PO2s that we have, or the higher partial pressures rather, the higher PO2, the higher PN2, which is the partial pressure of nitrogen gas, the higher the pressure we're going to see exerted on those surfaces. So what we end up seeing, or what we see encountered in kind of normal physiology of of the pressure of oxygen is that we see that as one increases their elevation, so as someone goes up in altitude, up into the mountains, up to higher elevations, the percentage of the gases in the atmosphere, as we stated, stays the same. There's just fewer gas molecules overall and lower partial pressures of each of those gases. So the reason why it's significantly more difficult to breathe at higher elevations is because there is less partial pressure of oxygen pushing that oxygen into the bloodstream, and as a result, there's less oxygen overall that's diffusing into the blood to bind to the hemoglobin.